All right, so in our previous topics, we covered how materials are transported in and out of the cells. Now, in this particular topic, we're going to talk about how large molecules or large substances are transported in and out of the cell. Endo meaning in and exo meaning out. Typically on your keystones, this refers to proteins. Proteins are very large molecules, and so they can't. Right, so if this triangle represents a protein molecule, they can't simply cross through the phospholipids, and if proteins want to come into the cell, they can't simply go through the phospholipids, which look like this, by the way. All right, they're just simply too large, and they just can't fit. So in order to transport large molecules like proteins, um, they need to get packaged into what's known as vesicles. So let's just go back and recap how proteins are made. All right, so proteins are made on these ribosomes. So I'll put the ribosomes in these as these blue dots. All right, the protein is, let's just say, is this red triangle. All right, a protein is made by these ribosomes, and they're going to get packaged into what's known as vesicles. Right, these vesicles will bud off right off of this endoplasmic. Let me label some of these things. So this is your rough ER. All right, that's going to package the protein. The protein is here in red. I'll label that too. All right, protein. Right, and blue, these are your ribosomes. That's where proteins are made. All right, so proteins are made on ribosomes. They're going to get packaged by the rough ER into these containers, and these containers are known as vesicles. And vesicles transport material. In this case, they're transporting proteins. All right, now this vesicle will go to the Golgi, right? And inside the vesicle is this protein. Right? So this vesicle will go to the Golgi. It'll get modified by the Golgi. All right, vesicle moves to the Golgi. All right, let me label that now. And then once that protein gets modified, it's going to get repackaged into another vesicle. Right? These are vesicles now. Right, this time packaged by the Golgi into a vesicle, and eventually that vesicle is going to merge with the plasma membrane. And once it merges with that plasma membrane, that protein is going to get released. Okay, and that is known as exocytosis. So this here is exo. Think of exit. So this whole process is known as exocytosis. All right, now the opposite can happen where, I'll draw this a little more simply. The opposite can happen where a protein needs to enter the cell. So here's cell number 2. All right, by the way, this is exocytosis, this process. And let's say this protein, and this one is, I'll just put an orange. No, why? Let's just keep it red. All right, so this protein needs to enter the cell somehow. And again, it's too large. So the same kind of process. This one is going to get absorbed by the, let me redraw this, by the cell membrane. Okay, put this arrow back. All right, so here's cell number 2, and it's going to form a little pocket. The membrane is going to form a little pocket, and that's where that protein is going to go into, or any large molecule, in fact, that needs to enter the cell. All right, now this pocket is going to turn into, what do you think? A transport container. What are these called? These are known as vesicles. All right, so the the cell membrane is going to butt off a vesicle, and that's going to go to wherever it needs to go. And I'm not going to draw everything else here again, but I think you get the idea. Large things that need to enter the cell this way, this one is known as, I'll do this in a different color, endo. When I think of enter the cell, cytosis. Okay, just completely reverse reactions. All right, endocytosis is what's going on right here.